Welcome to this presentation on cell division. It is intended as a preparation for physical lab work or as a review of concepts after the lab is completed. Cell division is the replication of the DNA of a parent cell and the distribution of that replicated DNA to the new daughter cells. Of course, the new cells also receive the cytoplasmic components of the parent cell. DNA is the molecule that controls cell function and contains the cell's hereditary information. DNA is located in the chromosomes. The way in which the cell divides depends on what type of cell it is. Prokaryotes, such as paramecium and bacteria, divide through binary fission, while eukaryotic cells are more complex and reproduce through mitosis and meiosis. Binary fission is simple compared to mitosis and meiosis. Prokaryotes have no nucleus and only one chromosome. This chromosome is a naked circular DNA molecule. During binary fission, the chromosome attaches to the plasma membrane and replicates. Cell growth lengthens the cell, separating the original and the replicated chromosomes. The plasma membrane constricts at the midpoint, forming a cleavage furrow that eventually pinches the parent cell into two genetically identical daughter cells. Cell division in eukaryotes is far more complex. There are many chromosomes in the cell nucleus, each consisting of a DNA molecule and its associated proteins. In cells not undergoing division, chromosomes are extended and uncoiled and not visible as distinct structures. During cell division, the chromosomes condense to form rod-shaped bodies that are visible with a light microscope. Chromosomes usually occur in pairs. Each member of the pair contains similar hereditary information. Thus, each pair consists of two homologous chromosomes, meaning similar in structure and genetic material. Cells with chromosomes occurring in pairs are called diploid. That is, two sets of similar chromosomes are present. When there is only one set present, the cell is called a haploid cell. We describe the diploid number as 2n and the haploid number as n, or half the diploid number. In eukaryotic cells, two different processes produce distinctly different types of cells. Cells formed by mitotic division contain the same number and composition of chromosomes as the parent cell. So a diploid cell that undergoes mitosis will produce two diploid daughter cells. Cells formed by meiotic division have only half the number of chromosomes as the parent cell. A diploid cell that divides by meiosis produces haploid daughter cells. Mitosis and meiosis refer to the orderly process of separating and distributing the replicated chromosomes to the new cell. Cytokinesis is the process of actually forming the daughter cells. Through the life of a cell, there are several recognizable stages. They make up what is referred to as the cell cycle. These stages can be grouped into two major groups, interphase and the M stage. The bulk of a cell's life is spent in interphase, beginning with an initial growth period, G1. Cells then move into a synthesis or S stage, in which the chromosomes and centrioles replicate. The final stage of interphase is a second growth stage called G2, which prepares the cell for mitotic or meiotic cell division. The M stage, mitosis or meiosis, takes up only about 5 to 10% of the cell cycle. While illustrations of mitosis typically show distinct phases, the process is a continuous one. Let's look at the stages of mitosis in greater detail. While a cell is in interphase, there is a distinct nucleus and two pairs of centrioles. Chromosomes are uncoiled and visible only as chromatin granules. During prophase, the nuclear membrane and nucleolus disappear. The chromosomes coil like a tight spring, and under a light microscope appear as rods. The centrioles migrate during prophase to opposite sides of the cell, and spindle fibers form. Each pair of centrioles and its astral rays form an aster or pole at each end of the spindle fibers, not unlike the magnetic poles of our Earth. In this fish blastula, we see the nucleus is less defined, and spindle fibers are becoming visible. There are two asters forming here and here. Metaphase is a very brief phase. Sister chromatids of each replicated chromosome attach to separate spindle fibers by their centromeres and line up at the equator between the two asters. This cell clearly illustrates metaphase. Anaphase begins with the separation of the centromeres of the sister chromatids. Once the chromatids separate, they become daughter chromosomes. So a cell in anaphase contains two complete sets of chromosomes. Once separated, the chromosomes move toward opposite poles of the spindles, as we see in this cell. In telophase, a new nuclear membrane forms around each set of chromosomes, producing two new nuclei. A nucleolus reappears, and the chromosomes begin to uncoil. Cytokinesis begins during the latter part of telophase and is marked by the formation of a cleavage furrow, and ends with the division of the parent cell into two daughter cells. Mitotic division differs slightly in plant cells compared to animal cells. The cells of most plants do not have centrioles. However, spindle fibers are still present. Cytokinesis usually, though not always, occurs during telophase. A plant's cell wall prevents formation of a cleavage furrow, so during cytokinesis, a cell plate forms to divide the two new daughter cells. Remember, the purpose of meiotic cell division is to create haploid daughter cells from diploid parent cells. Therefore, the process differs from that of mitosis, in that meiosis consists of two successive divisions but only one chromosome replication. 4 haploid cells are created from the diploid parent cell. Each of the 4 contain only one member of each chromosome pair. This process also reshuffles the genes, hereditary units formed in segments of DNA, and this significantly increases genetic variability among the daughter cells. Let's begin looking at meiosis in animal cells. Because there are 2 successive divisions in meiosis, the cells go through the phases of meiosis twice. So we refer to the phases with the specifier 1 or 2, such as prophase 1 or prophase 2. In prophase 1, each chromosome is composed of 2 sister chromatids joined at the centromere. The replicated members of each chromosome pair join in side-by-side pairing called synapsis. Chromosomes in synapsis are often called tetrads because they consist of 4 chromatids. Exchange of chromosome segments frequently occurs between members of the tetrad, increasing genetic variability in daughter cells. In metaphase 1, synapsed chromosomes line up at the equatorial plane and attach to spindle fibers at their centromeres. Anaphase 1 begins with the separation of members of each chromosome pair. The centromeres do not separate, therefore each chromosome still consists of 2 chromatids joined at their centromeres. Members of each chromosome pair migrate to opposite poles of the spindle in their replicated state. Telophase 1 proceeds to form a nuclear membrane around each set of chromosomes. The chromosomes untwist and the nucleolus reappears. Cytokinesis separates the parent cell into 2 daughter cells. At this stage, the nucleus of each daughter cell contains only one member of each chromosome pair in a replicated state. Thus, each daughter cell is now haploid. Between meiosis 1 and 2, these new daughter cells go through interphase, in which the centrioles replicate, but not the chromosomes, which already replicated prior to prophase 1. Prophase 2 is characterized by the typical loss of nucleus, spindle formation, and the appearance of rod-shaped chromosomes. Metaphase 2 sees the chromosomes lining up. Each chromosome consists of two sister chromatids joined at the centromere, which is attached to a spindle fiber. Anaphase 2 begins with the separation of the centromeres. The sister chromatids, now called daughter chromosomes, move toward opposite poles of the spindle. Telophase 2 proceeds as usual to form the new nuclei, and cytokinesis divides the cell to form two haploid daughter cells. Because each cell entering meiosis 2 forms two daughter cells, a total of 4 haploid cells are produced from the original diploid parent cell entering meiosis 1. This can be summarized as follows. One diploid cell enters meiosis 1, forming two haploid daughter cells, each of which enter meiosis 2, forming two more haploid daughter cells for a total of 4 cells. This concludes our introduction to cell division. I hope you found the presentation helpful. Thank you for watching.